All right. We're switching gears. And so everything we've talked about so far is, what do organisms need to grow? Like, what are nutrients they need to grow? What's everything they need to grow? And then, how do they grow once they've got all those nutrients? Let me get rid of that thing on the screen. And we're going to switch gears completely. Instead of everything on how can we get them to grow, now it's all how can we stop them from growing? How can we kill them? How can we at least just make it so they're not growing anymore? So chapters 11 and 12, I apologize, I think it prints on your thing as chapters 9 and 10. Chapters 11 and 12 is all about controlling or stopping microbial growth. And so it breaks it down into the two chapters, which is today's lecture and Monday's lecture. Today's lecture, chapter 11, is how do we stop bacteria from growing like out in the environment, on tables, on different surfaces. And chapter 12 is all about how do we stop bacteria from growing in the body, because it's two completely different ways. If I want to stop bacteria from growing on your table, I can spray it with some bleach. I can spray it with some Lysol. If I want bacteria to stop growing in the body, we are not injecting bleach. We are not drinking bleach. Like there's totally different ways on how we treat in the body versus out in the environment. So two different chapters. Maybe it's not that. It's just this. So starting with chapter 11 is all controlling microbial growth in the environment. And yes, the picture on the very top right should look familiar because, yes, even using incinerators, we're stopping bacteria from growing on your loops when we stick them in the incinerator. So in this chapter, we're going to go over what are some terms when we're talking about microbial growth, some considerations on, well, which one do we choose? Which antimicrobial are we going to choose? And then we group it into physical methods of controlling microbes from growing and chemical methods on controlling microbes from growing. So there's a lot of terms on there. This comes right out of your textbook. I was going to say, I know your textbook has one similar. It's chapter 9 textbook. Same things in the table. The biggest things I want to point out for some of these terms is because you've heard of some of these terms. You've heard of sterilization, sanitizing something, pasteurizing, disinfecting, germing, aseptic, antisepsis. And you're like, well, aren't they all like the exact same thing? I mean, they all are related to controlling or stopping microbial growth. Some of the biggest things are to note is sterilization. The big thing with sterilizing is you are killing everything. If we sterilize something, there is nothing growing. Even the hardest to kill organisms, dead. They are gone when we consider something sterile. If we say, oh, well, we sanitized that, because that is used quite often, especially like in restaurant settings. Oh, well, we used a sanitizer. Well, we sanitize this. Sanitize doesn't kill everything. It kills most things. It's not the same as sterilizing. The idea is when you sanitize, you kill most disease-causing organisms. Most. But it's not the exact same as sterilizing. Ultimate goal when we're in the medical facility is really to try to get to a sterilization level. If you go around and just wipe everything down with Lysol, that's not sterilizing. That'd be sanitizing. You're going to kill most disease-causing things, but you're not at that sterilization level. So since we're all talking about how do we kill these things, well, how do these things kill? We know if we put bleach on something, it'll kill. We know if we stick it in our incinerator, it'll kill. We know if we put some Lysol on it, we know it will kill. But there's two, and you're like, well, how does it kill? There are two main methods, no matter what kind of antimicrobial method you use. There's two main ways that these different things kill the bacteria. One is they do or damage their cell wall, or they damage some type of protein or nucleic acid. If you damage the cell wall and or the cell membrane of an organism, it's like if you knock down all the walls in the building doesn't exist anymore. That's the idea. If we damage the cell wall or cell membrane of a bacteria, the bacteria is dead. It's no longer able to grow and survive. If you damage the proteins, an example are enzymes. Enzymes are proteins. Proteins are made for structural material. They're made of, used for enzymes. Organism can't grow without the proteins. If you damage the nucleic acids, that's the DNA or RNA of different organisms. If you damage that, organism can't grow. It doesn't have the blueprint to make more of itself. So all of these different things are trying to target either the cell walls or the membrane, or they're trying to target proteins or nucleic acids on the inside. And that's how they kill. Now, how do you choose different antimicrobial agents? Well, these are things that we try to say we would like all of our antimicrobial agents to meet these 4 things. We want them to be cheap, because no one wants to spend $50 on a bottle of Lysol. We want our things cheap. We want them to work super fast. We don't want it to, okay, well, I got to spray Lysol, but it's not actually going to do anything for, 36 hours. Like, we want it to work really fast. We want it to be stable, which means we can just stick the can of Lysol in the closet and ignore it forever. So we don't want these things. They have to be under some type of, watchful eye, because it could explode or break down. We want it to be stable, shelf stable for a long period of time. And we don't want it to hurt anything. We don't want our different types of chemicals or different agents. We don't want them to hurt us. We don't want it to hurt animals. We don't want to hurt the objects that we spray it on. We want it to only harm the bacteria or the virus or whatever it is that we're trying to kill. So that's like our ideal agents. We want them cheap, fast, stable, and harmless to everything except what we want to kill. Now, things that we need to consider when we are trying to think, okay, well, what do I want to grab? Do I want to grab the bleach? Do I want to grab the Lysol? And these are just 2 common examples of things that we can use. Well, we do have to consider, well, what are we actually treating? Are we spraying something that's going to be interacting with an animal? Are we spraying something that is going to be interacting with, human touching, that we don't want bleach on something, then we're going to touch our skin on it right away? Are we cleaning, wood versus are we cleaning metal? I'm like, what are we actually treating? We do have to consider the site that we're treating. I'm like, there's certain things you can spray on metal that you wouldn't spray on plastic, because plastic could degrade down where the metal might not. So we have to, what are some considerations? I put a pet up there. It's not even my pet. I used to have a picture of my dog. It was a while ago. But, what are some considerations? Just what are things that have to interact with the site that we're testing? Also, are there any environmental factors that we have to consider? Are we spraying something that's super hot out and it's really hot outside? Are we spraying a surface but it's really cold outside? Is there pH that might affect our different chemical that we chose? So we also just have to look around, what are some other things? Is it in the sun? That'll break down different chemicals a lot faster if we're spraying stuff outside for disinfecting. So there's a lot of things we just have to consider. We can't just say Lysol will always work in this exact way when the temperature could play a factor, UV light could play a factor, wind could play a factor. There are other things that can play a factor when you're choosing different antimicrobials. And then there's, what is it that you want to kill? Since we have a lot of different antimicrobial things, we do have a lot of different ways to kill organisms or stop them from growing. We also have to what level do we want to actually kill our organisms? Do we know we're working with something that could actually be quite deadly and it's hard to kill, like those endospore bacteria, the C. diff that can make endospores? So there are different levels of disinfectants depending on what things we want to kill. And so we group it into our levels of different disinfectants for high levels, intermediate levels, and low level disinfectants. A high level disinfectant, and I'm going to go into examples of these at the end of this chapter. The high level disinfectants are going to be the ones that are going to be used to kill the hardest to kill organisms. And for most common practice, it's those endospore bacteria. So high-level disinfectants, these are going to be the really strong bleaches, or if we actually have sporicidal agents, these are going to be the things that are going to be used if we know we're having a C. diff or some other type of Clostridium outbreak of something. An intermediate level disinfectant is going to kill some of our gram-negative bacteria. Again, they are generally harder to kill because they've got 2 cell membranes and a cell wall that these different antimicrobial agents have to get through to be able to cause damage. They're going to be able to kill some protozoa as well. Don't get to protozoa to one of my last PowerPoints of the semester. but they're a eukaryotic organism that causes disease. Harder to kill, so you need an intermediate level if you're wanting to kill these types of things. Low-level disinfectants are going to kill the easy-to-kill stuff. A lot of the gram-positive bacteria, non-envelope viruses, even some of the envelope viruses, they're easier to kill, so they don't need a high-level disinfectant. We don't need a sporicidal agent. We don't need a strong bleach. Your basic Lysol, your hand soap, I was going to say, it's not hand soap, but just your basic hand sanitizer is generally a low level, sometimes intermediate level, depending what's in it, disinfectant. And like your hand sanitizer is not going to kill endospores. Now, the types of control, and I'm going to put up like there's two groups of types of control. There are physical methods on how we can control growth, and there are chemical methods on how we can control growth. So there's physical ways on how we can kill or stop organisms from growing, and then there's chemicals, because I've been going up in a lot of chemical examples so far with Lysol and bleach and alcohol. But there are physical ways we can stop organisms from growing, and some of these we do already in the lab. So one physical way we can stop microbes from growing is changing the temperature that the organisms grow. One is by increasing the temperature. When you increase the temperature above their happy 37 degrees Celsius, you're going to denature proteins. You're going to melt the cell membrane. The cell membrane of any of the cells are made up of phospholipids. If you increase the temperature, you melt the membrane. And so when we increase the temperature above that 37 degrees, our bacteria are not going to be growing. And if you increase it enough, like on an incinerator, you're just going to fry those things. Now, because there are multiple different ways we can warm or heat these things up to stop growth, we do group the different types of heat-related methods into moist heat and dry heat. And if you're a person that hates the word moist, I apologize for today's PowerPoint because I'm going to say it a bunch. So we group these different heat-related methods, moist heat and dry heat. So moist heat. So again, this is going to be using any type of increase in temperature, but there's also going to be some type of water involved as well. Boiling would be an example of moist heat. And before we had any type of autoclaves or really even knew about bacteria, we did know that if you boiled things, it seemed to kill stuff, or people got less sick. So before we had different types of sterilization methods, they're like, well, just, boil those clothes or boil the whatever that we needed. We need boiling water for everything. Boiling does not kill everything. It will kill most organisms, but not endospores. So your C. diff and other Clostridium organisms can survive boiling. Boiling is not a sterilizing method. It would be a sanitizing, and so it will kill a lot of stuff. Now, does anyone know what temperature water boils at in Celsius? 100. I wish everything was always just easy. So water boils at 100 degrees Celsius. And again, won't kill everything. Kills a lot, won't kill everything. So one thing to help us increase how much we can kill is we do what's known as autoclaving. And that's why we've got a big old autoclave that's right by your lab coats upstairs that hisses and beeps and all that stuff up there. The difference is with an autoclave is we increase the pressure, which allows us to increase the temperature. So by putting our objects that we want to sterilize, because it gets to sterilizing, in there, if we increase the pressure that's in there, we can actually increase the boiling temperature, and we can get the temperature up to 121 degrees Celsius, so above boiling temperature. And if we can maintain that higher temperature under pressure, because it needs the pressure to get up that high, for 15 minutes, autoclaving is a sterilization process. And it's why we do use it upstairs, because we sterilize everything before it gets tossed or before we use it for something. Anyone can? What do you can? was lazy this year. I gave all my tomatoes to my mother-in-law, and she can't salsa. What do you can? Only because I'm like, we're going to talk about canning, because there are different canning methods. Do you guys boil or do you pressure? Both. I was going to say, boiling kills a lot, but it doesn't kill everything. But there's a reason why you can boil can versus pressure. When you pressure can, you're autoclaving. That's the idea, is you bring it up to pressure to get above the 100 degrees Celsius. The biggest difference when you boil can, this is all fun factoids, to use outside of microbiology. When you boil can, why you can boil can stuff is when you boil can, is you can boil, say the same words. You can boil can things that have acid in them. Because between a boiling temperature and for things that are slightly acidic, that will cause it to kill everything. That's why a lot of fruits that have natural acids in them, you can can. Tomatoes have a natural acid to them, which is why you can boil can them. When you get into certain vegetables, or if you ever can meat, they do not have acid in them, which means if you do have to boil can, you have to add a lot of lemon juice. That's why a lot of lemon juices are in recipes, is that adds the acid. That's why you can boil can stuff and pressure can other things. So, just a fun factoid. But pressure canning is autoclaving. Showing that, yes, we have a huge autoclave upstairs, but that's not the only kind of autoclave. There's a ton of autoclaves. We've got several of them in this building that are just small little tabletops that, again, you put things in it. This is like the one upstairs. Whatever you want in there, puts everything under pressure. There are steam valves and pressure valves and all of these other things, which is why you always hear lots of pressure releases and hissing and beeping and stuff like that. And then there's a computer that monitors that it actually did get up to that 121 degrees Celsius, and it maintained that for 15 minutes. And there's a computer screen that will give a readout that proves that. But then we don't always believe the computer. Sometimes, not sometimes, all the time, we're going to put some type of indicator in the autoclave that indicator will indicate that it actually got to that level for that amount of time. This is showing one that's in a glass vial, that if we autoclave it, it's called an endospore strip, meaning if you can kill endospores, you've gotten to the sterilization method. And if it turns yellow, that means it worked. If it stayed pink or red, it didn't work. You, for whatever reason, didn't get up to that temperature for long enough. I thought I had another picture. These vials are kind of putsy, and so there's actually tape. It looks like masking tape. Then you just put masking tape on everything you put in there. And there's something they add to the masking tape at the outside that when it gets to the correct temperature for the length of time, there's a chemical reaction that happens on the masking tape that it will show a different color. Sometimes they're fancy and will read out autoclave. Some they just turn colors. But there's going to be some indicator. We never just believe even the computer readout because you want to make sure that things truly are sterilized if you're using autoclaves. Another type of moist heat is pasteurization. And pasteurization is used for, I don't want to say sterilizing, depending on which pasteurization method. But pasteurization is for when you are trying to kill things in a fluid. This is not something if you're, trying to sterilize some instruments or some type of tubes or anything like that. Pasteurization is if you are trying to kill organisms in a fluid. So pasteurization was initially used for making wine and beer. Yes, we use it for milk all the time now. It's because we're trying to kill all the things that are in there. Now, pasteurization, again, is not sterilization. So are there microbes in your milk? Yeah. Just buy milk, it has to be refrigerated, and it doesning last forever. The idea is, again, most stuff is killed. Again, we are heating it up, which is why the pasteurization process can change some of the proteins in the milk. And so they say unpasteurized milk can taste slightly different than pasteurized. Yep, unpasteurized milk comes straight from the cow, and the number of bacteria that's in there. Has anyone ever had unpasteurized milk? Can you taste the difference? I was gonna say, I'm like, some people do. What? Yeah, I'm one of those, I've never done it, even though I'm like, I grew up, like my grandparents had dairy cattle and all that stuff. Never done it, and I'm like, I don't think I would anymore, only because my husband does work for Quick Trip, and they do get raw milk in, so that they test it before it goes to the pasteurization process. And even he's like, oh, the stuff that grows, he's like, it's just ridiculous. He's like, I don't know why anyone ever drink raw milk, because you think fecal matter everywhere with cows. They try to clean everything, but it's fecal matter everywhere. But stuff's in your milk. It can make it through the pasteurization process. Again, the idea is that it kills most things, but we still have to refrigerate milk. However, there is something called an ultra-high temperature sterilization pasteurization process. And this is where we can actually pasteurize milk to the sterilization level. And we can do it, it's under pressure, that we can actually get the fluid up to 140 degrees Celsius. And if we can do that even for 1 to 3 seconds, it will kill everything. And it allows you to have milk shelf stable. Have you guys ever drank milk that's shelf stable? You can buy it at the store. I was going to say, I think some of the muscle milks, and you can just buy them right off the shelf. They don't have to be refrigerated. Anything that has dairy in it, I know even some of your canned coffee drinks, they have milk in them. They're not in the refrigerator. These are all things that were sterilized using those ultra high temperature. This is used quite often. For us, we're just like, oh my God, we can't drink milk off the shelf. But in other parts of the world where they don't have refrigerators, they don't have electricity that's reliable, grocery stores don't have electricity that's reliable, most of their milk is shelf stable. But once you open it, you expose it to things, and then it has to be stored in a refrigerator. We're just this weird country that, we just have to buy our milk in the refrigerator. You don't have to. You can go right to, I was going to say, I bought it before, because you can buy chocolate milk in little tiny cartons that's not refrigerated. But you can buy it at Woodman's and any of the grocery stores. You can buy it. Look for it now. Ultra high temperature sterilization that was done to it, though. Away from moist heat, because I had to say it one more time. On to dry heat. So dry heat is generally used when you are sterilizing or sanitizing instruments. And it allows us to get to higher temperatures for longer periods of time. We can get above just 140 degrees Celsius. One of the most common ways of dry heat is just incinerating them. That's why we have incinerators in our lab. There's no moisture whatsoever. It's really high temperature. We're going to flip it away from heat, because it's more moist. Gotta throw it in. There is more moist heats versus dry heats, temperatures, but we still can use temperature to decrease microbial growth, is by decreasing the temperature, by using refrigeration and freezing. Now, anytime you lower the temperature, it will decrease microbial metabolism. It's just like as it gets cold here, we all slow down. Well, same idea, cells slow down as it gets colder. So they slow down their metabolism, they slow down their growth, and they slow down their reproduction. But it does not stop any of those. Refrigeration and freezing does not kill bacteria. Just heads up, doesn't kill it. You can't throw something in the freezer, you're like, that'll kill everything. Nope, you just made organisms just go really dormant. All the time. That's why we've got one in our lab, is we pull stuff out of the incubator and we put it in the refrigerator, and the idea is that it just slows microbial growth almost to a standstill, so that when you guys look at your organisms, they look exactly what they look like when we pulled them out of the incubator. If we want to freeze our organisms, there's 2 ways we can freeze organisms: slow freezing and quick freezing. And part of the reason for that is, what do we want to do to our organisms? One of them is more deadly. Doesn't mean it will always kill the bacteria, but one's more deadly to bacteria than others. So slow freezing, which is usually what happens when you put something in your freezer, because your freezers aren't that cold. It allows the water to slowly freeze. And as it does that, it can form little ice crystals which actually is more deadly for bacteria. Won't kill them all, but it will kill more of them. So freezing can kill some bacteria, but it's not going to say it's going to sterilize everything. Quick freezing is when we use things like minus 80 degrees Celsius freezers that are way below that freezing temperature of 0 degrees Celsius. When we quick freeze things with a really cold temperature, that water freezes so fast it doesn't allow those sharp ice crystals and spikes to form, and so the bacteria are allowed to survive. And this is what we want to do sometimes when we want to preserve organisms for long periods of time, such as up in our labs. So we have minus 80 degree freezers to be able to really quick freeze those organisms. They don't die, we can pull them out, we can study them, play with them later in our labs. So one's a little more deadly, but not completely deadly, using your freezers versus quick freezing. Physical ways we can stop bacteria from growing. That doesn't include temperature. One is desiccation, which is just the really fancy official word for, say, drying. Bacteria need water. They're living organisms. They need water to grow. If we deny them their water, they are not going to be growing. Now, if it's a bacteria that can go into an endospore state, it can do that. So just drying things out is not going to sterilize, but it will inhibit or stop most microbes from growing, which is why if we want to preserve foods for a longer period of time, we dry them out. We dehydrate foods. When we remove the water, the microbes are not going to grow as well. This is why, again, before we knew anything about bacteria, we knew that if we dried meats, they would last longer. We knew that if we dried fruits and vegetables, they would last longer. They would not spoil, and it's because we're removing that water that inhibits microbes from growing. And then we have something called lyophilization, which is freeze drying them, which again, we remove the water first before we freeze them. And this is if we want to keep the microorganisms for long periods of time. We can freeze dry them. Generally, we don't ever freeze dry anything in our labs. We don't need to keep them for decades and decades. If our stuff dies, we just buy some more. But the CDC, if they do want to preserve something, we can freeze dry it. If we ever freeze dry any kind of foods, that's going to be growing as well. Can also stop microbes from growing. It's a physical method, is we just remove them. We can filter things out. So this is why if you ever have, I think I have more pictures. Thought I had a picture of your Brita filters, things like that. That physically removes them so that they can't be in that substance to be able to grow. This is just showing an example that there is a filter on here that we suction the water through by pulling it through, but all the microbes, all the bacteria are going to get stuck on the filter paper and they can't make it through, which means now your fluid should be sterile if you've got a good enough filter. I don't know if I'd say for sure Brita filters would sterilize. I'm curious to test it. Another way we can filter is not just filter fluids, we can filter air. We do have really fancy things called HEPA filters that we have these up in the hoods that we work with upstairs when we're working with microbes, and obviously we don't want to be breathing in anything, that we can get the microbes going up and filtered through. You can buy HEPA filters for your homes now that will trap microscopic organisms, little type of microbes. The idea is to hopefully trap bacteria and viruses that could be in the air. The HEPA filters, it is like its own brand name, and it's because they use a lot of, I don't say electricity, but the idea is that the electrical differences, they trap the bacteria by causing positive ions, so attract negative ions, and it traps them in the filter. And so they're really effective at actually sterilizing or getting pretty close to sterilizing the air. So we use them in our labs, but you can buy them in homes now. Another physical method is using osmotic pressure. And anytime we talk about osmotic pressure, we're talking about osmosis. This is water going where there's not any water. So again, it still has to do with kind of that dehydration process. Is if we add a lot of salt or sugar to something, it draws water out of the cells. So if we put a cell in something that's got a lot of salt or a lot of sugar, it draws water out of the cells, and that could be drawing water out of the bacteria cells. So that's what's happening in a hypertonic solution, just means it has a lot of salt or sugar. And when you put bacteria in a high salt or sugar environment, it draws the water out of the cells to the point that they're not going to be happily growing anymore. And so we use salt and sugar to preserve things. A lot of our meats, to preserve them, we just pack them full of salt. Ham is notorious for having lots of microorganisms in it. So when we ever pack ham of any kind, whether it's just your deli meat or if you buy a big old thing of ham for Easter, it is just like soaking in salt. That's why ham is so salty, is that they put it in that hypertonic salt solution to try to inhibit any type of microbial growth. It's also why, like if you can things, a lot of foods, fruit things, jellies, we just other than canning them, we pack them full of sugar, and sugar is also going to inhibit a lot of microbes from growing. Now, putting things in high salt or sugar is going to inhibit a lot of bacteria from growing because, again, it's going to draw that water out. Fungi don't really seem to care. Fungus can grow in high salt, high sugar environments just fine. They can withstand that loss of water and are still going to grow. But at least high salt, high sugar content is going to decrease bacterial growth. It's also why, again, beef jerky, not only is it dehydrated, it was like jam-packed full of salt. Not just for the flavor, if you want to eat something salty, but it does decrease the chance of microbes from growing, and it allows it to sit on the shelf for like a year or two. Well, that was my hypertonic, the little cell shrivel. Another physical way we can stop microbes from growing, using radiation. Two types of radiation, ionizing and non-ionizing. It just has to do with the wavelengths. Ionizing, the wavelengths are shorter. Non-ionizing, the wavelengths are larger. No, I can't remember. Non-ionizing radiation, an example of it would be UV light. And UV light is very commonly used. It's becoming, in the last 5 to 10 years, so even right before COVID happened, they were realizing the benefits of UV light and non-ionizing radiation in the medical field, that certain patient rooms were not only just getting wiped down clean, they would bring in UV lights to then also sterilize it. Because they know that this UV light does actually kill a lot of viruses, including COVID. One thing now, and I'm like, and you look at it the next time you go into a Walmart, right next to the carts, there actually is a UV light. I'm like, Walmart is trying to take care of you guys. Not that I shop there. But next to the carts, they do have a UV light next to those carts, and that UV light is there to help kill things that are on those carts. UV light cannot penetrate into something, but it can kill something that's on the surface. So if you're just trying to kill microbes on the surface of carts or on the surface of a bed or walls or whatever, UV light is great. And so it cannot penetrate into something, but it's great for killing stuff on the surface. Ionizing radiation, because it has smaller wavelengths, can actually penetrate into things. And ionizing radiation is really not commonly used on humans because we don't want that radiation to get inside of us, but it is commonly used on our fruits and vegetables. And so before your strawberries ever get to the store, before your raspberries ever get to the store, these are fruits that have lots of sugar that some bacteria do like. They got lots of fluid and moisture and nutrients in it. Microorganisms are going to grow like crazy in a lot of different fruits and vegetables. But before they get to the store, they actually will run them through a whole factory where they get radiated. And that ionizing radiation can actually penetrate into the fruits and vegetables and kill any microorganism, fungus, bacteria, whatever might be in there. And they're going to be able to sit on the shelf for a whole lot longer. Fun fact. We do irradiate our fruits and vegetables. Haven't heard anything if it tastes any different. Now, of the microorganisms that we do work with, and we do try to consider, high levels, low levels, because I didn't know where to throw in this. When we work with these different levels of microorganisms, we call them biosafety levels. So this is us working with the different levels of microorganisms. Biosafety level 1 is if you were ever working with organisms that generally do not cause disease in healthy humans. So we're assuming people that have working immune systems. They are not going to cause disease. And pretty much everything we work with in our lab, I'm going to say, or just about, I've got to start the next one. We work with in our lab, they're not going to cause any disease to you guys because you guys all, as far as I know, have healthy working immune systems. Our next to biosafety level number 2, because these are a couple of the organisms we work with up in our lab, meaning they can be moderately hazardous, meaning they can cause disease even in healthy individuals, but these are not going to be deadly diseases, but they can make you sick. Example, we are going to be working with a bacteria that causes strep throat. You guys, as long as you're not licking the plates, you're fine. But even if you have healthy immune systems, you could still get strep throat. It's not going to be deadly, but that doesn't mean it can't cause disease. So we do work with some organisms in our lab that are biosafety level number 2. We do not go anything above that in our lab because we just don't need to for our class purposes. But organisms that are people that go into the medical field that have to identify these different organisms, the med lab tech people, they are going to have to work with some higher level organisms because patients are going to come in with anything under the sun. So biosafety level number 3, these microbes have to be stored and worked with in safety cabinets. You have to work in hoods. You definitely are going to have a higher level PPE than just your basic gloves and your basic lab coat. Biosafety level number 4, I would be, they're rare, and I'd be shocked if anyone would ever be working with these guys, but you take proper precautions anyways, even in our labs here in La Crosse. These are going to be things that if you work with them, if you do pick them up by working with them, could be severe or fatal. My example is in the CDC. This would be a worker that's working with the biosafety level number 4 organism, which is Ebola. We're making a comeback. That yes, if the person would pick it up, it is quite deadly. And we will talk about Ebola when we get to viruses. Exactly. We're not working with any of those guys. That's cool things. Now, switching to chemicals, which is a little faster than going through all the physical methods. Again, the biggest thing is that when we are adding a chemical to an organism, we are trying to damage its cell wall. So it affects the microbe's cell wall, cell membrane, proteins, or nucleic acids. So we're trying to add a chemical that's going to affect one of those part of the microbes. We again still have to consider environmental conditions because bleach is going to work different in different temperatures. It's going to work different in different humidity levels. But we do have chemicals that are effective against all microbes. So we do have chemicals that can sterilize. Now I say review different kinds on your own, but I'm going to flip through them because I don't think there's really anything you have to write on the slides. But just a quick overview of different chemicals that we can use. Phenols and phenolics, these are the most commonly used things in the healthcare setting, in labs, and in homes. This is going to be some of your basic hand soap, your Lysol. And so they're commonly used in households, they're commonly used in healthcare settings. And they are intermediate to low level. Your hand soap, your hand sanitizer, different hand sanitizers have different things in it, but your hand soaps, your Lysol, it's going to kill a lot of stuff, but it is not going to kill everything. And again, it just works to denature proteins and disrupt cell membranes. Another one, your alcohols, which most hand sanitizers are alcohol-based, not all of them though, and they also work to denature proteins and the cell membrane. They did test that alcohol hand sanitizer is more effective than just using soap and water at the sink. Obviously, if you can do both, it's even better. But killing-wise, alcohol can kill better than just soap can. It's one of the reasons why even before any type of injection is done, they use a little tiny alcohol wipe right there before they do a shot. The idea is to kill most things that might potentially be there so you don't poke them into the skin. Intermediate level, Again, it's used, they damage enzymes by denaturing them. Widely used, you can tell, one common example of a halogen is iodine. We do have actually iodine tablets. Have you ever traveled to anywhere where you know you're not going to be able to drink water? You can actually put iodine in it. It will kill most things that are in the water, and you can drink low levels of iodine. It's fine. So I'm like, we do have different types of iodine that can be put on skin. It can be ingested. Different types of chlorine and Clorox are also considered halogens and are used quite often in pool water, used in hot tubs. Some added to water. Depending on the day of the water, if you turn your water on, you could actually smell almost a bleachy smell to it. Hot water's had chloride added to it. Oxidizing agents, this is when we get into high-level disinfectants. And so these are going to be killing the endospores. They kill by oxidation. They're using toxic forms of oxygen. Examples, hydrogen peroxide can kill. It's a very high-level disinfectant. Ozone, that's O3. Peracetic acid are all oxidizing agents. You know, fun factoid, ozone, which, you know, oxygen gas is O2. Ozone is O3. But that's not my fun factoid. The water in lacrosse, if I turn that water on, all the water in lacrosse that goes through the water treatment facility, because I've taken a tour of it, other than tons of filters and adding chlorine, the last thing they do before they send the water out of the building is it all goes through ozone. And it's literally meant to kill just about anything and everything. So that's why I say, depending on where you get your water, tap water a lot of times is actually safer to drink than bottled water, depending on how they've sterilized it. Surfactants, these are low-level disinfectants that are commonly used. Their job is just to reduce surface tension so that the bacteria can't stick to surfaces as well. A lot of soaps and detergents are surfactants. So yes, just when you wash your clothes and you're adding some detergent to it, that's not sterilizing. You know, you throw it in the heat in the dryer. That heat probably kills more than the detergent does. A lot of sprays, I'd say it's not Lysol, but it's similar. A lot of sprays that are sprayed on things. They're called quats or quatzels. It's based on the structure. They're just a low-level disinfectant. It disrupts cellular membranes. Heavy metals are low-level disinfectants. They denature proteins. Examples of some heavy metals that have been used to stop or control microbial growth: silver nitride, thimerosal, or copper. Has anyone heard of thimerosal before? I'm going to say, this is one. It is a heavy metal, and they know that it is antimicrobial. And 20, 30 years ago, I can't remember what the exact date was, they stopped using it. 20, 30 years ago, thimerosal was used as a preservative in vaccines. And so the anti-vaxxers are like, oh my God, you're giving us heavy metals, and of course vaccines cause autism. 500 times. One, thimerosal has not been put in vaccines in the last 20 to 30 years. There is no link to thimerosal ever causing autism. But to make all the anti-vaxxers happy, we took it out 20 to 30 years ago, and they're still saying that it's in there, and it's not. But that's the only reason I've ever really heard thimerosal. It's not used anymore, at least not anything that would be in vaccines or given anything to humans. But it is a metal, and it was very much used as an antimicrobial agent. Aldehydes, high-level disinfectants, examples: glutaraldehyde, formaldehyde, more safely known as formalin. If you've ever been, in your high school science classes, you probably all had the jars that just sat on a shelf somewhere. And you're like, why is that frog still in there? 1950, whatever. Well, it was preserved in probably a formaldehyde or a glutaraldehyde. High-level disinfectant, which means there's nothing else growing in there. And if there's no fungus in that jar with the frog, if there is no bacteria, virus, nothing, if there's nothing else in that jar, there's nothing to break down whatever organism you put in there, which is why you can preserve those things forever. I don't know how long they say that you can preserve them. Usually they fall and break apart. Formaldehyde, so I have formalin. Formaldehyde, which was one of the most common used preservatives for things, they found out later, oh, well, that can cause cancer, because everything goes in these days. So they've changed it, they've changed the chemical makeup. It's now called formalin. It's the non-cancer causing version of formaldehyde. But, 30 years ago, I want to say it's about 20, 30 years ago, they started making the switch from formaldehyde to formalin. Gaseous agent, there are certain things that you just can't spray with a fluid. If you've got tubes of something, you just can't spray fluid inside of tubes anymore to make sure it actually comes into contact with every single part of an inside tube. So we do have gases that we could put tubes inside of a gas chamber, and we know that the gas will get everywhere inside that tube to try to actually sterilize medical instruments, because a lot of medical instruments are tubes. Even the tubes when you give blood, well, you need to make sure all those are all sterile. So those are all put through some type of gaseous agent. So it's used in hospitals and dental offices, but really nowhere else. It's medical facilities only. Main reason for it and why this is not something you go to the store and you're like, I want to start using gas to kill everything, because gas is hazardous, explosive, poisonous, and some can cause cancer. So that's not commonly used. We would never commonly use it, but medical facilities, dental offices that have to make sure certain tubing is sterile, yes, can get it put through gas. And there's different types of enzymes. We talked about enzymes last time, that they can break stuff down. Well, we know that, and so there's certain enzymes we can actually add to the organism to try to break them down. An example, lysozyme is an enzyme that breaks apart the cell wall of bacteria. So we can add enzymes that can target certain parts of bacteria. We also know there's an enzyme that can target prions. We'll talk about prions when we talk about viruses. They're really hard to kill organisms. And they'll kind of make the news, usually right around this time of season, because chronic wasting disease in deer is a prion disease. So as antimicrobials themselves, they're just used to treat disease. They're used outside of the body. But the problem is, the more we use of antimicrobials, the more opportunity we give these organisms to figure out how to work around it. So the more we expose organisms to soap, the more we expose them to hand sanitizer, the more ways they're like, oh, well, what if I try this? Nope, that didn't work, I died. And they sit there, and the more opportunity you give them to try different things, the more opportunity you give them to eventually figure out a way to survive different types of antimicrobials. So the idea is we need to use antimicrobials. We know that, but we don't want to overuse antimicrobials when we don't need to. So there's that fine balance of we want to kill them, but we only want to kill them when we want to kill them. And that's where I'm going to end, because we're going to talk on, it's not chapter 11, it's chapter 12. On Monday, we're going to talk about all the different antimicrobial drugs. So all the different types of drugs, your penicillins, your ampicillins, all those different types of things. How do they kill bacteria? I group these two chapters together always because they talk about the same type of topic, just where you take them. There is not a separate online quiz. So I looked, I'm like, there is, I was going to say, you've got to take the test if you haven't taken it. I didn't look to see who's taken it already yet. But there is a test due tonight by midnight, if you haven't taken it, about the metabolism PowerPoint and microbial growth. That's due tonight, but then there's nothing else that's due until I see you guys on Monday. So you can have a good Oktoberfest.